Hi everyone, back to channel. Today let us see the Rickettsiaceae. So first understand the general characters of this. It was mainly honored, its name was got because of honor of Taylor Ricketts. So you can see he is a Howard Taylor Ricketts scientist who worked on the spotted fever and died of the typhus fever. So he got he was honored for this. And these are the small gram negative bacilli. Remember that they are gram negative and they are obligate intracellular parasites, that is they can live only inside the cells and transmitted to humans by arthropods except the Q fever. So infections are manifested as fever, rash and vasculitis. So they are grouped depending on the clinical features, epidemiology and other characters such as cultural characters and vectors. So let us understand about the family Rickettsiaceae. Rickettsiaceae, so family Rickettsiaceae, genera Rickettsia, Orienta, Coxiella, Ehrlichia. So these are the different genus, and the species are the Provezeki, Typhi, Rickettsi, Conori, Ostris, Sebrechia, Acari, and so Orienta, Coxiella, Ehrlichia. So let's see here the overview. These are obligate intracellular bacteria, that means they can grow only within cells. They are the agents of typhus, spotted fevers and also the Q fever. So what are the biological characters? So first let us understand its morphology. So they are small gram negative pleomorphic cocobacilli and their size about 0.3 mu to 1 to 2 mu, here you can see, and they are non-motile, so they can't move, and non-capsulated, they doesn't have any capsule, you can find them, and they're very short rods actually, and they're not stained with the gram stain but visible under the light microscope when stained with the Gimsa, Gymnus, Castenda and also Machiavello. So the Castenda and the Gimsa give them purple, while the Gymnus and the Machiavello give the red stain. So they also possess a trilaminar cytoplasmic membrane and also a cell wall. So the cell wall resembles the gram negative cell wall, and they possess both the RNA and DNA and divide mainly by the binary fission. So these are the important points. You can see they are stained poorly, positive by the Gimsa staining. And let us understand the cultural properties. So the Rickettsiaceae are obligate intracellular organism because they are unable to produce sufficient energy. Therefore, Rickettsiaceae must be grown in the cell culture, embryonated eggs or experimental animals like mice. And in embryonated eggs, we take the yolk sac, and Rickettsiaceae divide by binary fission within the host cell. And we need to know some other important points. Actually these Rickettsiaceae, they do not grow in a cell-free cultures, so we need to grow them example in the cytoplasm, that is the typhus group, and in cytoplasmic vacuoles, especially the Coxiella group. So the yolk sac, we should put them in at least 5 to 6 days, especially the old chick embryo is used, so they multiply in the cells of the membrane surrounding the yolk sac. And the cell cultures, they grow in many continuous cell like HeLa, HEP2, Detroit 6, mouse fibroblasts, but are not satisfactory for the primary isolation. So Rickettsiaceae grow well in the cells that are non-metabolizing. This is very important. And the animal inoculation can be done not only in the mice, many people say mice, but one more important is the guinea pigs. So you can remember it. And the factors for the survival of this Rickettsiaceae are: this can't survive longer outside the vector or the host, so they're quickly destroyed by heat, drying, disinfectants like hypochlorites, ethanol, 1% of Lysol and also 2% formalin. So the dried fecus of the infected lice may contain this infectious form for months at the room temperature. So the Coxiella may survive the pasteurization and also in the dried milk for the months. So we need to be careful with the lice fecus. We can't see it properly, can't understand whether it's not, but always be careful. So always hygiene is important. And the antigens present in the Rickettsiaceae, especially the group specific soluble antigens, are present on the surface of all organisms, and also species specific antigens are present. And alkali stable polysaccharide is found in some Rickettsiaceae and in some strains of Proteus like OX19, OX2, OXK, so which forms the basis of the Weil-Felix reaction, which is used for the diagnosis of the rickettsial infections. So let's understand the outline sketch of this, how it infects our body. So first it enters into our body by a bite of an arthropod vector or through their fecus. So next it enters and undergoes the local multiplication in the endothelial cells of the small blood vessels, and next it enters the blood and next damages the vascular endothelium and causes the disseminated intravascular coagulation, that is DIC. You know what happens in DIC? The blood clots in a certain place, and as well as because of all coagulant factors are involved there, the blood on the other side gets so too leaky, because whenever an injury it gets too leaky, it is so at the same time it has coagulation and also has too leaky because of absence of coagulation factors in other side, and also other hemostatic disturbances are taking place. So what are the diseases caused by this? Mainly the typhus fevers, the spotted fevers, and also the scrub typhus. So what are the various features? Let us understand. So there are many diseases as we discussed, and next we have the laboratory methods. So before going into the laboratory methods, first let us see the culture property here. Rickettsiaceae are obligate intercell. Okay, we know this. Unable to, therefore Rickettsiaceae must be grown in cell culture. Oh, okay, this we are completed. And coming to the pathogenesis. So as we discussed, they are maintained in nature in certain arthropods like ticks, lice, fleas, mites, and transmitted to humans by biting of an arthropod, infecting primarily the endothelium of the blood vessel, as I discussed. So following a bite by an infected arthropod, the organism are taken into the human cells, like you can see flea, louse, mice, so mite. So these are the things which are causing this. And clinical finding, as I said, typhus fever, spotted fever. So damage to the vessels lead to the typical rashes, fever, severe headache. In addition to headache, the DIC shock because of in DIC as blood is leaking, so hypovolumic shock because of low blood in the body, and also other profound central nervous system changes such as coma can occur in severe cases, because if blood is lost, even it may lead to coma. And next let us see the laboratory methods. Actually we take the specimens by blood and also biopsy from the trash. So method of detection, we can detect it by the direct detection of the Rickettsiaceae or antigen. So it can be done by the smear prepared from the biopsy of a rash stained with the Giemsa and also Gymnogen stain, and also we can do the direct immunofluorescence test. So in the nucleic acid detection we can use PCR, and in isolation we should attempt it only in the laboratory with safe provisions, and can be achieved by animal inoculation or either by yolk sac. If you remember animal inoculation, we already discussed it. So blood clot ground in the skimmed milk or brain heart infusion broth is used in male guinea pig or mice animal, observed for 3 weeks, and that response is called Neil-Musser reaction or tunica reaction. So animal develops fever, scrotal inflammation, testes cannot be pushed back into abdomen because of adhesion between the layers of tunica vaginalis of animal, and it varies in different infections in the animals. And in Rocky Mountain spotted fever, that is RMSF, it is caused by R. rickettsiae, and it is causing the fever, scrotal necrosis, and also animal may die. So in spotted fevers, it is mainly caused by R. typhi, R. conori, R. acari, where the fever and tunica reaction are common. And in epidemic typhus, it is caused by R. Provazeki, that is causing fever, no testicular inflammation here, remember. And in scrub typhus, it causes O is by O. tiskus gamshi. A mouse is preferred, becomes ill, develops ascites, smears from the tunica, spleen shows the rickettsia. So yolk sac, this also can be used for the growing Rickettsiaceae, and also we can grow Orienta. So what are the serological methods? It is mainly the Weil-Felix test, and also we use the specific test using Rickettsiaceae antigens. So this is all about that. And coming to the principles, prevention and treatment. So before seeing this, we also need to see about the Weil-Felix test, because it is important. It is an heterophile agglutination test which detects the antibody in the patients of a rickettsial fever that cross reacts with some strains of protease due to sharing of the alkali stable carbohydrate antigen of Rickettsia and also O antigens of a non-methyl strains of protease. So and also P vulgaris, that is OX19, OX2 and P micro OXK. It is non-specific and insensitive, that you need to remember. And the procedure is the serum of patient is mixed with this OX19, OX2, OXK antigens in a tube agglutination test and observed for the agglutination. And interpretation, the tests are interpreted by different diseases like epidemic typhus, Brill-Ginser's disease, endemic typhus, tick-borne spotted fever and scrub typhus. And specific tests are like complement fixation test, specific but insensitive, and direct fluorescence antibody test, enzyme immunoassay, latex agglutination, for RMSF positive in acute infection, so diagnostic should be done. And how this can be treatment prevented, last point, based on the reducing exposure to the arthropod, wearing protective clothing, removing the arthropod, maintaining good personal hygiene. Of course, there is no vaccine and other preventive measures, so only be careful, no vaccine treatment. Carry on the antibacterial treatment, that is mainly antibiotics. So let us know how it can be treated, mainly by sulfonamide, that is enhances the severity of the disease, and sensitive to tetracycline and ciprofloxacin. So remember, sulfonamide is enhancing the severity of disease, means it is resistant to it, and sensitive to tetracycline and ciprofloxacin. So we recommend ciprofloxacin and tetracycline for this. This is all about the Rickettsia.